Have you ever noticed how similar siblings are and yet how different? Or pondered why people do the things they do, vote a certain way, love a certain guy, prefer eating salads, even get cancer? And whether these things are preordained by our genes? Well, Tim Spector has, and he's spent 20 years looking for the answers. Brilliantly, he chose identical twins to study, real-life clones who start out the same, get the same upbringing, and yet turn out in reality to be very different, even in the way they die and when they die. It is a privilege to welcome Dr. Spector to the Museum of Science and to have the opportunity to hear about his fascinating research and what he knows now about how we become the individuals that we are. But before we dive in, Dr. Albright would like to say a few words, and I'd like to introduce him. Please join me in giving Dr. Niall Albright a warm hand.
Thank you, Lisa, and thank all of you for being here on a very special evening for the Albright family and for the museum. This has been in the plans for over a year. When Jim Krause came to me and he said, what about putting everything together, put all the interests of the Albrights together and see if there could be a symposium that would introduce and honor the Hall of Human Life. This is a terribly exciting idea to me. Our family has benefited so much over so many decades of what has happened here at the museum. And to me, as a scientist and a surgeon, if there's excitement when you're teaching science, it's a winner. It's going to make a lot of children interested in it, and a lot of children are going to go into it. Myself, at age 15, I got involved with this museum, believe it or not. As a Milton Academy student, I was invited to come to the museum by Brad Washburn, my hero and mentor. He gave a presentation, and by the end of the evening, he had accepted my great desire to be part of the next expedition going to Mount McKinley. We went to Mount McKinley for 3 months, researching it in a way only Brad, who was known in Alaska as a true pioneer, or what I called him, a real McKinley sourdough. He was absolutely magnificent and in his element, both photographically and in proving the object of our expedition. And that was to prove who was the first person, after great controversy, to climb Mount McKinley. And indeed, we traveled so many mountains, so many tents, so much of Kilton Glacier to discover both the true peak of Mount McKinley and some 17 miles away, what we called the fake peak that Frederick Cook had claimed was his first ascent of the tallest mountain in North America. Life magazine developed a 6-page centerfold article about this, and to this day it's influenced me along with the tent companions, 6 others who were on this expedition. To this day have been mentors for me. When Jim Krause said, why don't we put the interests of the Albrights together into something that would build some excitement well before the opening of the Hall of Life, and when he said it could honor people that have gone before and done so much for the museum, I was truly excited and jumped for it on 2 conditions. Number one, that it would focus and interest everybody in the Hall of Human Life, but that it would also honor my mentor, Brad Washburn. He agreed to this, and it has taken place indeed with the help of Lisa Monrose, with Liz Kong, with Lucy, with so many people of the entire museum staff. We now have before us a wonderful adventure, a wonderful step forward in exciting people about their own health and human life. So it's with so much pleasure that I turn this podium over to now a good friend and colleague from London, Tim Spector, to talk about genetics and genes to us tonight. Thank you for coming.
Thank you very much. And I'd like to start off by thanking the Albrights and the museum for giving me this amazing opportunity to come to Boston and talk to you on this very special evening where I know I'm going head-to-head with the game of the century. So hopefully we can wrap up and you can still see the winning runs on your way home. But I do appreciate it, and it's great to be here. Darwinian dogma tells us that it's impossible to change our genes. But many of us, looking at our parents, might wish that wasn't so. Now, I know my kids in particular do. But what I want to do is to explore that more, but also to explore what makes us individuals, and that we are more than just a bag of genes. Now, before I start, I want to gauge the audience with a little quiz. And I'm going to tell you why afterwards, but I just want quickly you to tell me which of these four figures here, 1, 2, 3, or 4, represents the number of different genes there are in the human body. All those, no conferring, no dialing a friend. How many think it's number 1, 23,000? How many think it's number 2? How many think it's number 3? And number 4? Number 4 wins. In terms of the numbers you think it is. The real answer was the few cognoscenti who knew it was number 1, 23,000. Okay? And for those of you who picked this group here, you have probably more in common with London taxi drivers than with London doctors. So most doctors in the UK that I've tested have no idea how many genes there are. And this tells us something about how fast this whole field is moving, so that by the time people went to medical school, and they just haven't caught up on their reading. So taxi drivers know more than doctors about genetics. That's the equivalent. So if you ever go to London, ask a taxi driver. He's bound to tell you all your answers you want. Now, I never asked worms the same question because I haven't got that communication technique down to a T yet. But I can tell you that the average human has very similar numbers of genes to a worm. So when you're thinking about how special you are, God's creation, etc., then just think carefully that maybe genes aren't the only answer. Maybe it's not the number of genes you have, but how you use them that's important. That makes us different to worms. So let's go to Iran. In southern Iran, about 6 or 7 years ago, and we're going across the desert, and there's a funeral cortege. And there are 2 coffins in this cortege, and a crowd that's full of mourners weeping as they bury these 2 small coffins. And in those coffins are these 2 twins who lived all their lives together, conjoined, what we used to call Siamese twins. And what's remarkable about these 2, Laden and Lale, is that although they shared all the genes in their body, and they had, for obvious reasons, exactly the same environment all their lives, until their early 30s, they were remarkably different. One was shy, the other extrovert. One was religious, and the other less so. One preferred computing, the other reading. And one wanted to be a journalist and the other a lawyer. And in fact, they so much wanted to be different that they were prepared to take the ultimate risk and a really risky operation to separate them. And it took 55 hours and 20 surgeons, and sadly it failed. And in the end, they got what they wanted. They were separated at birth, and they did something on their own. Now, what's really interesting is, why would those 2 have those different personalities when everything we know, we're taught from science, shows that shouldn't be the case? And that's what I'm interested in. And for the last 20-odd years, when I started the UK twin register, I've been looking at thousands of twins, and for the most part have been trying to work out what makes them so similar. Why are identical twins more similar than non-identicals? And nearly everything we've studied has told us that everything we look at has a genetic basis. So they're more similar in the identicals than the non-identicals. And that goes not only for diseases, but it also goes for preference in foods, whether you like salads or fruit, whether you like garlic, and how strongly you believe in God. So there's nothing that you can measure accurately that doesn't have some genetic component, except which football or possibly baseball team you support. And I'm still working on that one. We might find the Red Sox gene. But when you scratch beneath the surface, you often, underneath those smiles, those personalities, the way they hold wine glasses or coffee cups, which are these natural poses, you often see more differences than similarities, particularly in personality and behavior. And you have to remember that these twins are clones. Okay, so when you think of cloning, this is what it's going to be like. Except you wouldn't have someone of the same age. But you have an identical egg, which after between 2 and 10 days splits into 2 identical pieces. And these are these individuals. So why should they be any different if the genes in every cell are the same? And this is what has really driven me in a lot of my recent research and started to change my view of genetics. So rather than looking at the similarities, I'm much more interested in these differences. And that's why about 3 or 4 years ago, I decided to work in this field and write a book on the subject. Now, we all have to die sometime. Some of us will die sooner than others. And there are some reasons why some of us people die earlier than others. Now, what's interesting in twins is that they live their lives together, and the expectation is that twins die at the same time. And if you take news stories, if you use the media as your guide to this, and like these friars who were from Buffalo and lived to the age of 92, died within a few hours of each other, you would think that's the norm. You couldn't be more wrong. Twins rarely die within months or even years of each other. And longevity is one of the least heritable traits that we've looked at, which is curious. Why would that be? And when I started digging in this, looked at our twins, and this slightly complicated slide just tells you that the lines in blue are showing what the similarity is in the identical twins. For if one has the disease, what are the chances of the other one having it? And you can see that for heart disease, it's about 30 to 40%, only about a third chance of one twin getting it when the other one does. And that's for a very common disease. For a disease like diabetes or rheumatoid arthritis, the risk is only around 1 in 10. These are amazing figures because we can't really explain that because these should be highly genetic diseases, and yet something else is happening. And what I believe this other thing is happening is epigenetics. This is the third element between nature and nurture. So we know that nurture, which is lifestyle and our environment, can influence nature via epigenetics. And epigenetics, quite simply, is the mechanism by which chemical signals can switch your genes on or off, or up or down, a bit like a dimmer switch, changing its function without changing the structure of the gene, and that this effect can last months, years, or possibly generations. Now, for the advanced scientists in the audience, this just tells you that epigenetics has lots of ways of doing that. These chemical switches are very complicated, and I'm not going to go into it, but the simple one is when methylation occurs. So you just get a methyl group, sticks on the top of a gene, and switches it off. But it can also, you can get these little signals which can unwrap your DNA or close it up, a bit like a lock. And so these little switches mean that your structural DNA can work in very different ways. And we know this is reversible, and this is what we mean by epigenetics. We're finding new mechanisms all the time. Now, one of the key experiments that happened about 6 years ago was done in the US, and you might be shocked to learn that they gave pregnant mums large doses of vitamins or alcohol. And they studied the effects on their children and on their grandchildren. And what they found was that those they'd given the extra amounts of either vitamins or alcohol to were more likely to produce children and grandchildren who were chubby blondes compared to skinny brunettes. You don't believe me? They're genetically identical sisters. The genes in these two animals are exactly the same, and they were actually brought up on a normal diet. And the reason for this is epigenetics. Now, what are some other possible examples that we might not have thought about? Now, for the last 50 years, rates of lung cancer have been dropping as smoking has been dropping. Now, these rates have come down faster in men than women. And in women, they're pretty stable and soon set to overtake breast cancer as the leading cause of death in most countries. But what's really interesting is that of the new cases, between 30 and 50% of women have never smoked. And we can't really explain that in terms of what's going on. So could it be that these kindly doctors in the 1950s who encouraged us to smoke certain brands of cigarette are just one of a generation where everyone smoked in the 50s and 60s, and that we're reaping, if you like, the side effects of that through these epigenetic signals? It also might mean that whatever we do in our lives could have an effect on our grandchildren in ways we hadn't really anticipated. There's some studies in humans that, from Bristol, that if your grandfather smoked before the age of 13, you're more likely to be overweight and be susceptible to diabetes and heart disease. And that's going 2 generations, even though you never smoked. And there's some other similar stories in animals, rats and mice, developing asthma. And could it be that because my grandparents smoked, I have a slightly increased risk of lung cancer, although I've never smoked a cigarette? Well, 1 or 2 maybe. Another example, what about these are rats, and rats like to be licked and cuddled by their mothers. Mothers are very loving of their rat pups, and that's the normal evolutionary way that rats are brought into the world. And if you deprive them of that, you take them away from their mother, then you're changing something hormonally in their brains and those chemicals. And it turns out that their grandchildren are much more likely to be anxious, neurotic animals, even if they've been brought up normally. Now, the good news is that it's partly reversible, that if you take those animals who've gone down that route and you put them with foster parents, you can reverse some of this, as you can with certain drugs that can reverse it. So this gives us a potential idea of failing families and children who are in broken homes, that possibly doing things very early and giving them back the nurture is going to chemically alter their brains and their genes in ways that are beneficial. So might have some implications for some of these policies about how long you leave failing kids in bad homes. So it looks as if the lives we lead might affect our children, our grandparents led could affect us. But what about pregnancy? Now, pregnancy is a key time when things happen epigenetically. There's lots of massive amounts of growth and development, and epigenetics are really important. Now, we know that women take lots of vitamins during pregnancy, and the number one is probably folic acid, which is also in the food supply in the US, not in all European countries. So getting plenty of folic acid, and that's really good for preventing spina bifida and other birth defects. Done a great job on that. But there's now evidence that this vitamin, which has a key role in epigenetics, it actually supplies those methyl groups that I was talking about. If you take too much of it, you can become subfertile, and there's a slight increase in cancer risk. So these things, once you start playing them artificially and taking very large doses, you can alter some of the things that are naturally beneficial. Now, even more worrying are those things we don't know we're taking, and women who are particularly pregnant taking. One of them is a toxin that epigenetically is like a weak poison. Epigenetically, it's called bisphenol, BPA, which is found in plastics. Now, I don't have to tell people in the US about how important plastics are in all our lives, and this bisphenol is in all plastic materials, and small amounts of it get into us and can affect us, and animal studies have shown that it can epigenetically alter neurochemicals in the brain and hormone levels. We know very little about it. Canada has banned it, but the US and the UK say, don't worry, it's safe. I'm not so sure. So we really need to worry about these things a bit more than we are doing at the moment. What else can we worry about? And what else could we think about that creates differences? One of them is something that I hadn't thought about until very recently when I started working in this area, and this is called the microbiome. These are the 100 trillion bugs that live in our intestines and on our skin. And it turns out they've co-evolved with us for millions of years, and our genome is made up of about 38% of these bacterial genes, which have got incorporated with us. So we can't really survive without them. And they've done experiments where they take sterile mice, and they don't live very long, and their brains rot, and they have no immune system. So we need microbes for our survival, and yet we're doing our best to destroy them, killing them off with antibiotics. The average 18-year-old has 20 courses of antibiotics in the US. And the other thing that's changing our evolution is cesarean sections, now the commonest operation. 30% of Americans are having cesarean sections instead of natural birth, and it's higher in, it's about 50% in New York and 60% in Puerto Rico. These are not for medical reasons. They're to get more time on the golf course for those obs and gynecologists. And it's cultural. But what's really interesting is that we're all born with no microbes. When we're in the womb, it's totally sterile. And as we're born, it appears that it's slightly random about which microbes get into us. And it depends on how the birth was done. And who actually touched us during delivery will determine the microbes you have for the rest of your life. And these microbes are important because many of them are associated with disease. All of us share about 40% of our microbes with each other. Don't take my word for it, wait till you get home to test it, but you're unique. And there's a reason for that, because genetically, we've done some twin studies showing that there is a heritable component. So a lot of these are due to environment and diet. What you eat will give you a different diet. And there's some evidence that high-fat foods are actually bad for your microbes, wipe out whole species that would normally be there and more diverse. So we're killing off a lot of our microbes with bad food. But genes are also important and control some of them. And some of those microbes are important in obesity. So you can actually grow up certain microbes that are either good or bad for obesity. They're either greedy ones or they're lazy ones. And not only in obesity, but also there's evidence that things like autism and brain effects can be altered by your microbiome. So lots of reasons why we're very different. And even twins are not that similar in terms of their microbiome. Now, going back to our cancer example I showed you earlier. So lung cancer actually acts epigenetically, as do most cancers. So as well as causing gene mutations, they can actually cause total disruption to our normal epigenetic mechanisms. And what they're doing is they're sending out these chemical signals which are switching off our natural defenses. All of us have thousands of genes on our genome that are there to prevent cancer. They're called tumor suppressor genes. And what happens is these chemical signals are being switched off, and that allows the little cancer cells just to start growing and metastasizing in their nasty way. And what happens when we smoke is that we're doing pretty much the same thing at lower doses. So the cigarette smoke is switching off exactly the same genes that the full-blown cancer is doing. So it's helping it along. But if we go back to our example, if you had your smoking grandfather, those signals could well be transmitted to us, much lower dose than actually smoking, but still switching these off in ways that just increase our risk a little bit. Now, the good news is, as I said, that epigenetics is generally reversible. Unlike gene mutations, which you can't switch back, these epigenetic switches are reversible. And if you stop smoking, you can, within 3 months, most of your epigenome returns to normal, which is good news. Some of it doesn't. It takes years, but a lot of it does. The other thing is that there are actually 4 drugs on the market in the US that are anti-epigenetic or antigenic agents used for cancer. Mainly used for leukemias, but there are 40 others in development, which are really exciting. So the idea is that you can actually reverse some of these effects with these drugs, and these are very much drugs you're going to see in the future. So it's very exciting. So what about what I've told you? I hope I've told you that by studying identical twins who are different, we can see that genes alone are not the important thing. Genes alone are not something that we can predict our future with. We can't use them for personalized medicine for the common diseases. Because if identical twins aren't very good at predicting each other, what good is a genetic test? It's never going to be as good as a twin. If you've got your nice clone to go with you through life, that's your best genetic test. So there's more to it. We need to be able to use these things, other things like epigenetics, like other scientific tools on top of the genes to be able to predict people well. And if you need any further encouragement, this is Cece the cat and her little clone. And the owner of Cece wanted her $50,000 back because, as you can see, adorable and cuddly, but look at the eye color, look at the coat color. Exactly the same genes, but a different cat. And that really illustrates to me epigenetics. You know, the devil is in the detail. And that's why we're never going to get the Gattaca-type scenarios in this area. Now, what can you do now? Going home, what take-home lessons are there? People like take-home lessons. Well, get on your bike, do some exercise. Exercising 3 times a week can reverse some of these bad genes you might have been dealt by your parents. The obesity gene, FTO, the #1 obesity gene, you can reduce its effect 30% by exercising 3 times a week. And there'd likely be other examples. Stopping smoking, we've discussed. Having a healthy, diverse diet, full of fiber, and also containing some of the vitamins that are key for epigenetics, such as betaine and choline, making sure you're not deficient in these things. Much better to take it with food than through vitamin tablets. Avoiding antibiotics, avoiding vitamins. I don't think they're good for you. And all the evidence is building up that epigenetic, they could be having the opposite effects to what you want, because you're getting large boluses of something that is not natural. Kiss and cuddle your kids as much as you can, because you need to switch on their empathy genes epigenetically. If they're not switched on, they're not going to treat you very well when you're old, grumpy, and incontinent. Really important. Go home and give them a big hug, even if they're 40 years old. We've talked about toxins such as bisphenol plastics. There are about 40 others on this list that epigenetically can be altered in minor ways that are now very difficult to measure, and many that are in varnishes and paints. And that's one very good extra reason that I don't do it yourself housework. Finally, whether you think you can or think you can't, you're right. Willpower has a genetic component and therefore has an epigenetic component. So use it, is my advice. And one of the messages we have is that we're all quite unpredictable. We may start with the same genes in life, but as we go through life and we have these effects on us through our lifestyle and environment and these genes pulling us in different directions, our lives will take different courses. And this is illustrated how twins go down different courses. And it also explains how we respond differently to the same scenario. And identical twins responding to the same stress of, say, being adopted or in a Romanian orphanage will often react in very different ways. So all of us can react to the same situation in very different ways for exactly this reason. So, in conclusion, by understanding what makes us individuals and very much different to each other, we can start to understand how we can change not only our destinies but also those of our grandchildren, while still remaining, luckily, identically different. Thank you. So, I'm very happy to take questions from the audience. You have to wait till a microphone gets to you, I've been told.
There we go.
I'm behaving.
Nicely done. First question right here.
So you mentioned that you have, we should eat fruits and vegetables that have vitamins that turn on the epigenetics. And you said something about betaine. Could you clarify that? I just didn't catch that, what you were referring to.
There are a group of foodstuffs that contain what they call methyl donors. And including those, these chemicals betaine and choline. And there's a whole group of these foods, and I'm not a nutritionist, but basically things like eggs and nuts and things like celery and certain root vegetables have large amounts of these in them naturally. So when they've done some experiments on animals, giving them these diets rich in these nutrients, and had similar effects to actually giving some of these epigenetic drugs. So at the moment, we're in the early stages. We don't know exactly the sort of perfect epigenetic diet. But what's generally clear from the epigenetics and also the microbiome is that the more diverse a diet we have, the better it is for us, and the more sure we are we're getting the full range of these nutrients. And my view is that taking them naturally is immeasurably better than going to the health food store and taking a whole multivitamin tablets.
Next question here.
Yeah, I was really interested in the heritability aspect that you mentioned, particularly with the smoking grandfather example. And I was curious, to what extent do you know that they were able to control for the causation factor in terms of, would this same factor that was inherited also predispose the grandfather to smoke, or how were they able to tell, that versus other lifestyle things that may have been correlated with the smoking? And also on the diagram there that you had of the three mothers in series, I think, I was curious in that vein also if you could explain what the arrows were. Thanks.
Okay, well, in humans, unlike animals, it's very hard to control for every factor, as you're probably alluding to. It's much easier in these laboratory situations to give a much clear-cut answer of what's going on. So the illustrations we have in humans are often historical ones. So we're not able to control for everything. We're just saying, well, let's look at the data and see what we've got. And that's why we rely on a couple of key historical things, and we haven't discussed this, but there are certain key famines in history that have been followed. So after the end of the Second World War, the end of the winter of 1944 in the Netherlands, when the Germans were retreating, there was a systematic starvation of the Dutch population. So 250,000 people were literally starving for 4 months over winter. And they were on about 600 calories a day. And they followed up those women who were pregnant and found that the next 2 generations were shorter and were more prone to diabetes and schizophrenia. And, there are other possible explanations, but what they compared that to other children born, say, just outside that window, 6 months later, and there was no difference in them. And there were similar famines in China, the so-called Great Leap Forward famines in the 1950s, where millions were systematically starved, and similar findings of shorter, more diabetic, more schizophrenic offspring. Data not very good, but in large numbers. And it's quite possible that the famines at the end of the American Civil War, predominantly down in the South, could have triggered changes that led to an increase in gain in weight. And that has persisted because as food supply has been very bountiful in the US, those signals have been the wrong signals. And that could have triggered the obesity. And if you look at the maps of the US, that's where the biggest concentration of obesity is. So all these have other possible explanations, but they do seem to fit these themes. And the one you were referring to, the smoking grandfather one, again, they looked at other possibilities. They looked and saw, well, if the father smoked much later in life, it didn't have as much an effect. And they adjusted for as many factors as they could, like smoking habits of the other parents, the rest of the family. They adjusted for everything they could. So they're never going to be proof in any human situation, unlike the animals. But my view is that if you can see these things in nearly all the animals that we've studied so far, the same processes are likely to be true in humans. But it is controversial, and the data, people will pick holes in the human data.
Next question's over here.
Is there any way that learning could be heritable epigenetically?
Yes, and there are some neuroscientists that believe that actually the formation of a memory, which is still highly debated, could actually be an epigenetic phenomenon because of the nature of the reversibility of it, that actually switching genes on in a very short time frame might itself have an epigenetic basis. It's obviously very hard to study that in brain, but that's one of the current theories. And the only studies I know are of mice that have been given rather nasty shocks in one generation. Getting food, they have to go through a little electric shock on their foot. And they've shown that their children and grandchildren are statistically more likely to be anxious doing the same procedure when they get food than in others raised differently. So they're very hard to do these studies, but I think it's quite possible that but we don't know whether we all have to have electric shocks in order to pass it on to our kids, or whether more subtle learning could be. But in theory, I think that's possible. And there are some other examples I think in the animal kingdom. Some people have suggested that elephants might pass on some knowledge of special places to hide and things epigenetically, but I'm not an expert on that.
Next question here.
One time I went to a Harvard Med School lecture for the public, and I got to ask a geneticist during intermission or something whether we are more like our siblings, like am I more like my sister, or am I more like my daughter? And she said, more like my daughter, because we have 23 chromosomes in common. But I don't really think that I quite understand that at this time. So I wondered if you could tell me why having the same parents doesn't make us as much the same as my daughter and I.
It's a complicated question and answer, but basically it comes to the fact that although both share 50%, both with your daughter and your sister, you share 50% of your genes. It's where they come from that's important. So you might share, in a way, the portion of your genes that comes from your father and your mother more with your daughter than with your sister. So it depends if those genes on which chromosome they're on. So it's an argument that is more theoretical than practical, okay? Because most of the studies have not shown any differences between mother-daughter relationships and sister relationships. But clearly, I, as a believer in epigenetics, think that probably you're going to be closer to your sister because the age effects and the cohort effects are so much greater. Your environments are going to be, in one generation, the environments are much more similar than all our kids who are going through completely different environments to those particularly of our parents. So we're at a time of such huge change in lifestyle and environment that our genes are sort of struggling to catch up.
Okay, next question's over here.
I have a question about the epigenetic drugs. Like, what is the efficacy rate as well as the endurance? One, people that take this drug, does it help everybody? And then lastly, if I go home and just eat a ton of choline, like if I stop, then does it actually start breaking down and, I lose the impact?
Good question. We don't really have all the answers, but basically the oncologists give these drugs in about 6-week cycles. So it seems to be, you don't have to have daily injections. They do seem to work in these cycles, but it's going to vary depending on the mechanisms you're looking at. So all the drugs don't just affect methylation. Some of them affect some of these other processes that I've mentioned, like the opening up of the chromatin and the closing. And we're only really discovering by trial and error, how these things are working. And it's quite likely that you can affect some of your epigenome every day, and other effects could last months or years. So there isn't going to be a one-size-fits-all. Okay, so you might take a ton of choline, and that's going to only work for a day or so in some of your genes, but in others, it might have a much more permanent effect.
Next question here.
I wonder if it's known or if you could explain how epigenetic, the sort of intergenerational transmission of epigenetic effects get into the gametes, actually, or do they, or are there other modes of transmission?
Another good, tough question. Up to about 4 years ago, we thought that when the egg and the sperm met, all the previous signals on those genes were wiped clean, a bit like a DVD that you just sort of reformat to start and record again. That was the view, because it would say, how can the body cope with all these old signals on there from an old person? We want to start again, a nice, fresh, young, single cell. And it turns out that actually, although most of it is erased, in the grooves of the DVD, there are a few little signals in there that tell you where it came from. And we don't understand the mechanisms, except that these are resistant to some of the wiping mechanisms that are there epigenetically that go on in the formation of that first egg. So, this is all very new, and we are slowly understanding. The key is to find out what are those signals that remain, and why do they remain? Why are they so crucial? Because nothing happens by chance. There's a reason that they remain there. And that's really, I think, some of the exciting work that's coming out now is to uncover what they are.
Okay, the next question's over here.
So you made a very strong statement about vitamins, and obviously it's better to get nutrition from a well-balanced diet. Are you actually saying, well, if someone doesn't have a well-balanced diet and cannot for some reason or has some deficiencies, that there's still more risk in vitamins than benefit, or what exactly are you saying about that?
No, I think clearly I'm not being that nihilist about it, and I'm still a practicing MD, and if someone is deficient in B12 or vitamin D or whatever it is, you give it to them. But I would always encourage people to take it from dietary sources first. And recently there's a controversy come up in osteoporosis, for example, about taking calcium. People say, well, you should take extra calcium, it's good for you. And it turns out that all the studies show that people who take long-term calcium are actually getting more heart disease because their vessels are getting stiffer. And and the calcium is being deposited in the vessels. Now, it's good for your bones, but not so good for your heart. So that's a good example of how the body may not be able to deal with large doses, boluses of these things that you wouldn't have to deal with normally. It's not natural. So that's my general point. But again, going back to the epigenetics, it's quite likely that some of these will also have other subtle effects that we don't know about by giving these very large amounts, like the folic acid balance, I said. Obviously, if you're deficient, absolutely, you need folic acid, but people taking these super doses looks really bad.
But what about non-super dose, just normal baseline multivitamins for people whose diet might not be at its best?
Well, I wouldn't advocate it, and I don't think we know the answer. Most of the big trials of multivitamins against non-vitamins show that actually people do worse, and they get more cancers. So, I wouldn't assume that just because it's a vitamin, which is all the PR campaigns say is healthy for you, it means it is. We need to do more work on it, but, let's change people's diets, not try and get it out of a bottle. I think that's the key.
Next question here.
Well, good evening. I enjoyed your provocative presentation, but I would like to ask you about the role of behavioral pattern versus, say, incidence of cancer. Because in laboratory animals, which are inbred and maintained under strictly identical conditions as much as we could do, that some strains of mice are very aggressive. If you work in the colony, you'll know that. And others are very placid. Some of them have extremely high incidence of cancer, and others very low. And I wanted to know how you deal with these kinds of big differences where the conditions are exactly the same in which the animal is living in terms of temperature, humidity, the food, the sterility, and the handling, and everything of this are quite as close to being identical as it would be, I think, pretty much feasible for Homo sapiens to generate.
So we're talking about genetically identical animals, are we?
Well,
Inbred strains.
yes.
So inbred strains, for people who don't know, are sort of as close as we can get to identical twins.
Yes.
They're not completely identical, but there's very little variation between them. And we do see this, and it's a well-known phenomenon that, yes, you get 20 of these animals. They will have outliers that behave differently. So how can we explain that with epigenetics? Well, we can explain it because epigenetics is a system by which we have a built-in variability, so that there are some actual genes that control areas of our epigenome that are likely to respond wildly to something, whether it's high or low. And it's the same idea of Romanian orphans who had terrible treatment and were locked away for 2 years in the dark, and then followed up 10, 15 years later to see what sort of teenagers they'd be. You can imagine most of them weren't in a good state. But 1/3 of them were untroubled. They were completely psychologically normal. And I think we have this built-in variability in humans and in animals that says that we're not always going to respond exactly the same way to every environment. And that is a protective device so that in situations like that, some will survive and some will succumb, but not everyone will be wiped out. So that protects the species. So I think we have this built-in individuality that gives us our own personalities in all of us, so that identical twins, when they're given, I spoke to some, and they said, they went to school, and they remember at the age of 8, sitting in a maths class, and down the front, and they decided that the maths teacher, one of them loved the maths teacher, one of them hated the maths teacher, and from that point on, one did science and one did art, and they just reacted differently. They were in the same class, everything was the same, but we have this natural sort of ability to act unpredictably, and that's really what I'm getting at, and that's a natural phenomenon. And it's chance.
But they're not living in exactly the same environment identically. I mean, they're exposed to different people, they have different images that they see or things that they hear, so they're not living under identical conditions, because everybody's neighbor environment.
No, but you can't keep everything, even in your laboratory experiment, nothing is absolutely identical. Airflows are going to be different. One day one might pick up a bug that the other one doesn't do. One steals one's food and one eats less. The way they were born, they come out in a different birth order. There's always some subtle difference between them. And I think we have to accept there is this built-in variability in all animals in the way they react to these responses.
These differences I talked about are huge between two different strains under very closely identical conditions, food and everything else.
No, I'm sure they are. I'm just saying, I'm agreeing with you, but I'm just saying I'm not at all surprised, and I can explain that because epigenetics is there. Some of these regions are so variable, the wind blows one way and these genes will be turned on. They're sensitive to be different. We're supposed to express ourselves in different ways, and all animals have this to some, because it makes sense. If every animal is exactly the same, they become predictable, they become prey, easy to kill. If you don't know if a mouse is going to bite you or not, that's, you're going to take a bit more care.
Okay, next question's over here.
Hi, is there a way to measure or sort of get an idea of our epigenetic makeup? Like I'm thinking with, you sequence the genome and so on. How do we understand our own epigenetic composition at any given time? I understand that it may change, but is there any approaches to measuring it?
Yes, well, in our twins, we're measuring 5,000 of our twins, the full sequence. So we're measuring, but to do that, we've got to look at 20 million points on the genome. Okay, so you get your DNA sample and you measure these sites, and at each of these sites, there's a chemical switch that's either on or off. And so you're getting 20 million sites to do that. Now, at the moment, I'm not aware of anyone offering that commercially. But it's not far away because, as many as you know, you can get 1 million of your SNPs tested now through companies such as 23andMe, which based in San Francisco, where by post you send off your DNA, a spit sample, and they'll basically segment your genome into 1 million pieces, reassemble it and tell you everything from your ancestry to your risk of having curly hair or baldness. Really important things like that. The rest of it isn't very accurate, but I was told I had 80% chance of having blue eyes. But I advise you all to go to that website if you are interested, and there are other ones, because it's fantastic value to learn about your DNA for $99, and that way you also learn about the whole process of how it's happened. If that can do that now, I suspect that within 5 years, companies will be offering epigenetic tests, which will say, well, okay, we know these are the important genes, and like for breast cancer, the Angelina Jolie story, you might remember, she had BRCA1 gene, which is this very rare, affects less than 1% of patients with it, and she had the high-risk one, and her aunt had died. She had this tested, but in identical twins, both with the same gene, only about 50% of them, 1 in 2 of them will get it. So it doesn't mean you definitely get it. So if you could add on top of that an epigenetic test to see if that gene was switched on or off, that would be really helpful. So I think these things are going to come in the future. The technology is still too tricky, and it takes one of my technicians, a year to sort out one disease. So it's going to be a while before it's as commonplace, but it will happen.
Next question here.
You had mentioned earlier that high fat has an effect on the genes. Could you explore that further for us? And how high is high fat? And the current diets are very high fat compared to former diets. Thank you.
Okay, well, my advice is only really going to be good for mice or rats in the audience. I know the cleanliness standards of the museum probably made that unlikely. So they've only really done these studies in rodents. And they always try to roughly equate it to a very high-fat diet. One of the studies claimed it was the average American burger diet was what these rats were getting. And they did show that by feeding these pregnant mums this American burger diet equivalent, they did find these epigenetic changes. But we haven't done the studies properly in humans yet to know what the right level is. We're just showing at the moment that with these extremes, you can switch genes on and off. The only experiment easily done in humans is smoking. So I can tell any of you by taking your DNA whether you've really ever smoked in the last 10 years, because I'll have those traces on your genes. And hopefully in the future we'll be able to see, well, have you ever had a McDonald's in the last 10 years? I'll be able to have a little switch on that.
Okay, next question's over here.
How widespread is both the belief and the research in epigenetics now?
It's still quite controversial because a lot of people who, in mainstream genetics, I think people accept that it exists. I think the controversy is how important is it? So I have some genetics colleagues, and they seem to think, oh, well, this is just what they call an epiphenomenon. Okay? It's sort of going around the problem. It's not really the cause of things. It's just something else that's happening. And it's because it's the way it's come. Some people, geneticists, feel a bit threatened by it because it's not mainstream genetics. It's not looking at mutations. It's not doing the classical studies. And it's come up with this rather strange route. But most people accept that, particularly in the cancer world, they know it's really important. They know that's how cancers work, that's how metastases work. So you speak to a cancer geneticist, there's no one who says it's not important. If you speak to the other area of disease, in diabetes or arthritis, people will be less sure. And there's still controversy about the ability to cross generations because it's so hard to do human studies. And the view is, well, if it happens in all other animals experimentally, it must happen in humans. It doesn't wash with some people. They think humans are so special.
Next question here.
So I'm curious. I want to have a mental model that my genes are my genes, and yet I know that the cells are differentiated through my body. When you study epigenetics, to what extent do you have to study a particular type of cell or a cell in one part of the body? To what extent do epigenetic effects migrate through the body? How uniform are they across a given cell type? And how does that affect the way you think about this?
Okay, that's a very advanced question. And absolutely on the money, because if you think about it, we all started life as one cell. And that one cell became divided and became over 200 different tissues. And each of those tissues, those cells all behaved in different ways. So they all had different epigenetic signals to make them produce proteins that made a difference between your tooth and your liver. but all inside each is identical DNA. So clearly there are marks that separate all the cells in all the tissues of the body. Now, what we've been doing in our work has been taking blood. We take blood, we're taking fat and skin, things that are on the surface and people have too much of. And what we show is that generally there's about a 70% overlap between the different tissues. So we know that some things are going to be very specific to a particular cell, but many of the signals we can pick up are general. So when people smoke, for example, you can see that in every cell that we can test. Very similar. When you look at age, you see the same age signatures in all the cells. So I think it's going to be a mixture. There will be certain specific that are specific to the cells, particularly function, and it's quite likely that brain cells are going to have quite a different makeup to other cells. But our hope is that by starting looking at the ones that are useful clinically that we can access, we can at least get 70% of where the action is and worry about the inaccessible ones later.
Next question over here.
Oh, go. So you had mentioned 4 drugs that were being given to cancer patients to help reverse what was happening and 40 others in production. I was wondering if any of those in production are meant to be preventative medicine as opposed to curing.
In general, drug companies don't like starting with preventive medicine because the bar is too high. They generally like to start with cancers or extremely rare disease or extremely fatal diseases. And that's why the field is working this way. So the only one I know of at the moment is one that's targeting obesity. So it's targeting some of the obesity genes that alter your appetite. So they're working as sort of appetite suppressants. That's what they're working on. But no, most of them are at the moment going for the obvious targets, the really blunderbuss approach. And then as they get more refined, they'll work it through. So that's generally the way any new drug class works, is you start with the most severe cases, and then hopefully once that's on the market, you then get other indications.
And this will be the last question.
Same question about.
Oh, it's the same question. All right, I got faked out. Who has? Oh, there we go.
I mean, I should say the vitamin. Obviously, there are many claims on the internet, if you look now, for special vitamins that have epigenetic preventive effects. But for the reasons I've said, we don't understand enough to know exactly what our balance is yet. And so, I think the best vitamins to take are as much fruit and vegetable as you can fit on your plate.
This will be the last question.
Tim, with these very exciting concepts and proof that you have so much from your identical children studies, I wonder, now that we've discovered the human genome in 2001, now that we're so aware of mutations, the intriguing question is, from your point of view as a geneticist and epidemiologist, how close are we and how can we get closer to gene therapy in humans?
Gene therapy has not delivered what people thought it would 10 years ago. And the reason is that, as we said, we have the same genes as a worm. And it's because they have so many different functions and uses that just by replacing a gene structurally, you're going to cause so many disruptions to things we can't understand. Because what we do know every year is that the whole story gets more complex. So we keep saying we know, we keep knowing 10 times more every 5 years, but we still only know 1% of what we currently know. We think there is out there. So I don't think gene therapy per se, in my mind, by actually replacing the genes, has a future. But I think our ability to understand the cell and how cell therapy, we can actually start to manipulate what those genes do on a much more targeted way, is something that we, hopefully I'll see in my lifetime. Thank you.